Lecture 3 is going to go over the three major functions of urine formation of the kidney. These are going to be filtration, reabsorption, and secretion. We've already talked a lot about filtration in lecture 2, which has to do with the glomerulus, but in this lecture, we're mainly going to focus on reabsorption and secretion. So urine is the byproduct of the balancing of the body fluids and removal of waste. So a lot of excess water and electrolytes are in the urine. That's actually what urine is mostly composed of. And then there's also a lot of waste in there as well. Glomerular filtration is the first step. So in that step in lecture 2, materials were moving from the blood plasma into that capsule. And then the contents in that capsule were moving into the proximal convoluted tubule. And that's where we're going to follow for this lecture, is that tubular system. Throughout the tubules, so throughout the PCT, the descending limb, the loop, the ascending limb, and the distal convoluted tubule, we're going to have a lot of reabsorption occurring. So it seems kind of counterintuitive where you have a lot of filtration happening and then we reabsorb a lot of those substances. But ultimately, it's going to depend upon what the body needs, what the cellular requirements are demanding at that time. So materials move from the tubular fluid back into the blood plasma. And it can do that because we have all of those blood vessels intertwining with those tubules. So we have the peritubular capillaries or the vasa recta intertwined with that nephron loop. And so those materials can move very easily back and forth. And then later down the tubular system in the distal convoluted tubule, we will have a lot of secretion, which is basically the removal of any lingering wastes that may still be in the blood. The glomerulus does a lot of it, but there are still some waste yet to be filtered. And so there will be a secretion mechanism going from blood plasma into the tubular fluid later in the nephron, and then that will be a lot of wastes going that way. So just to recall, remember that we have an arterial, a capillary bed, then a venule, right? And so the filtration in the capillaries is driven by blood pressure. And so that filtration happens in the glomerulus. And then water follows solutes. So that's something I want you to keep in mind for this lecture as well. Just a reminder on what filtration looked like in the glomerulus. There are two capillary beds. There's one here in the glomerulus and there's one here in the peritubular capillaries or vasa recta. So things are filtered out here in from the capillaries to the capsular space into the tubular system as filtrate. And then later down that tubular system, this would basically overlie this. This is just a simplicity drawing. But essentially, we have things that are reabsorbed from this tubular system that goes back into the bloodstream. But then we also have wastes that are left in the bloodstream that need to go into the tubular system. So basically, there's this constant exchange going on throughout the whole nephron. And it's quite complicated. So let's talk about reabsorption. So a lot of small solutes are filtered out of the blood in the glomerulus into that capsular space. But what if we need some of these substances? So let's just say that we get rid of most of the sodium in our bloodstream and it goes into our tubular fluid, but our body is requiring some sodium because our blood plasma is low in sodium. What we can do is reabsorb some of that sodium. So some of that sodium can move out of the tubular fluid, go back into the bloodstream. So a lot of these ions are transported through different mechanisms: simple diffusion, facilitated diffusion, which are passive processes. There's also some active transport, which requires ATP, and then water will move by osmosis. So following concentration gradients. The capillary blood is under relatively low pressure though, so it's very permeable. And so the plasma proteins that are in the bloodstream basically are attracting water via osmosis. Because of that colloid osmotic pressure, but the rest of this can pass mostly by passive mechanisms and some active mechanism as well. This kind of is a cool picture. This is all the vasculature of the kidney. So I'm not kidding when I say that a lot of the cardiac output is in the renal system at any given time. So again, reabsorption is going from tubules to vasculature. So into the paratubular capillaries or vasa recta, depending on what type of nephron we are. And the point is to have those capillaries throughout the nephron is to have oxygen drop off in the tissue, right? So just like any tissue, oxygen needs dropped off, carbon dioxide needs picked up and carried back to the heart and then out to the lungs. But also we need to transport solutes and water in order to make urine either more dilute or concentrated. Dilute would mean it has more water in it than solutes. Concentrated would be more solutes than water. But also regulating renin and those other hormones that we're going to talk about in lecture 4. So a lot of solutes are filtered out in the glomerulus. About 65% of those are actually reabsorbed in the proximal convoluted tubule. And kind of like the digestive tract, the proximal convoluted tubule has little microvilli that increase surface area for absorption. One thing that's highly reabsorbed there would be sodium chloride, and that is an electrolyte. So when this is divided into its individual components, sodium has a positive charge and chloride has a negative charge. And so they will actually create an electrical gradient that other ions will follow. Passively. Now, glucose is a fairly large molecule. It is transported via active transport. Amino acids, potassium, a lot of other things are reabsorbed in the proximal convoluted tubule. Again, the amount that's reabsorbed just depends upon how much is in the body at any given time and what the needs are at that time. About 70% of sodium is reabsorbed in the proximal convoluted tubule. So let's just say most of that is filtered in the glomerulus. About 70% will actually go back into the bloodstream right away, and 97 to 99% by the end of the nephron. So we retain most of the sodium. That's beneficial because, again, negatively charged ions can follow sodium. Water follows sodium. A lot of those things are going to follow it. So if we reabsorb sodium, we're also going to reabsorb a lot of other substances. It's initially moved with its concentration gradient in the glomerulus. So it's filtered out via filtration, but also with its concentration gradient. So it's going to go into the capsule. Now, then it's reabsorbed via against its concentration gradient via a sodium potassium pump. The energy from that is used for secondary active transport of other molecules. So sodium kind of leads the way and then other things are going to follow, or a couple molecules can go through the same carrier protein. Let me go over this an example here. Okay, so primary active transport basically creates a concentration gradient of sodium. So here's sodium inside the cell. It's going to move outside the cell against its gradient because there's more sodium outside the cell. Potassium is going to move the opposite way. That requires ATP. Well, the energy from this actually allows other molecules to basically hitch a ride with sodium going through the next carrier protein. So here, for example, glucose. So glucose is transported via secondary active transport. So as sodium moves back with its concentration gradient, right, from low to high, glucose is going to hitch a ride with that as well. And that's called co-transport. So there's co-transport here, and there's that electrochemical gradient. As sodium moves out, chloride will follow. As sodium moves in, chloride will follow. Bicarb is another negatively charged ion that might follow sodium as well. So here's a picture from your book. So here we have about 70% of the sodium, we'll say, was filtered out into the filtrate, into the tubular system. Let's say our body needs some of that sodium back. So we're going to reabsorb by active transport. And because sodium's moving that direction, it's positively charged, bicarb is going to follow. And then because these solutes are going into the bloodstream, now water is going to follow. And chloride is like sodium. So chloride is going to follow wherever sodium goes. And so that's going to follow as well. So not only is sodium being reabsorbed, but that will trigger this cascade of other molecules being reabsorbed as well. So you're probably thinking, what's left, right? We're going to get there. So the interesting thing about reabsorption is only so many solutes can be reabsorbed. And that is just due to transporters reaching their maximum threshold of how much they can carry. And so when we talk about saturation for receptors, right? So we only have so many receptors that can allow sodium back in, their bicarb back in, things back in. And once those things are all tied up or they're all busy, right, they cannot reabsorb anymore. And so there's only a certain level of reabsorption that can happen. If that threshold is reached, then we have things being excreted in the urine that maybe shouldn't be there, like glucose. Say we have a ton of glucose in the urine, in the tubular system. We reabsorb some of that glucose, but we reach our maximum amount that can be reabsorbed. And somebody who's diabetic has a lot more glucose than that in their body. And so glucose will end up going out into the urine and never being reabsorbed. But when you find glucose in the urine, that's an abnormal finding. So some things are normally found in trace amounts. You might have a little bit of bicarb in your urine, something like that, but some are not normal, protein, glucose, things like that, white blood cells, that kind of stuff. And when we have non-absorbed solutes, right? So we don't have a lot of solutes reabsorbed. Well, that means water is going to move into the urine to try and balance that, right? Due to diffusion, concentration gradients. And so then you're going to urinate more. So diabetics, they urinate more because they have more glucose in their urine, because not all of that glucose could be reabsorbed and utilized in the body. And so because there's a lot of glucose in that tubules, the water will follow, and then you're going to urinate more often. Now, not all wastes are filtered in the glomerulus. Some are left in the plasma and need to be secreted later. So now we're in secretion. So that was all reabsorption. Now we're in secretion, which is the removal of wastes from the bloodstream into the tubules, maybe things that pass the glomerulus, past that checkpoint, that first checkpoint. So these things are going to be transported into the tubule system by active transport. A lot of this is going to occur in the distal convoluted tubule. The distal convoluted tubule does some reabsorption. It does potassium, but it also allows hormones to take action. We're going to talk more about those hormones in lecture 4, but the hormones that directly control body fluid composition in the kidneys, that's where those things are going to kind of take an effect. One of the major things that's secreted from the bloodstream is going to be hydrogen ions. So one of the functions of the kidneys is to maintain body pH, and pH is driven by the concentration of hydrogen ions. The more hydrogen ions you have, the more acidic you are. And so sodium is reabsorbed. So hydrogen ions are going to be secreted. So those are going to go from bloodstream to tubules and be excreted in the urine. And your urine is slightly acidic because of those hydrogen ions. A lot of drugs and toxins are also secreted. And then potassium is also secreted due to the reabsorption of sodium. Potassium is going to move the opposite direction. So we'll have some potassium in the urine as well. So here's our distal tubule here. So we have some sodium moving here. We've had some reabsorption of sodium. If sodium is being reabsorbed, then potassium is going to be secreted. You'll also have the waste secretion here as well, ammonia, things like that. All right, so composition of urine as we go here. A couple images from your book. Let's start with this one first. So the composition of the filtrate leaving the glomerulus is basically isotonic. So there's the same amount of solutes and water here. Now, in the proximal in the descending limb, that is where a lot of water will be reabsorbed into the bloodstream. So if there's a lot of water being reabsorbed and there's still solutes in here, right? So we have a high concentration of solutes, that fluid is now hypertonic in the descending limb around the loop. In the ascending limb, now we have the reabsorption of some sodium and some chloride. And so now, because we have had some water leave, but now we've had a ton of solutes leave. Now we're hypotonic, which means that we have a lower concentration of solutes and a higher concentration of water. So that's kind of how the composition changes throughout the nephron. And this is all dependent upon the sodium chloride concentration in the kidney tissue itself. And so that is basically recycled. A lot of the NaCl is actually doesn't go very far from the bloodstream. It's constantly being reabsorbed and filtered out constantly in the nephron. And so here we have NaCl being absorbed here, water will go out, and then over here we have NaCl, some NaCl going out here. It's just following a concentration gradient based upon the interstitial fluid, which is that tissue fluid around this area. So here's kind of an overview table from your book. So basically it starts with filtration. So in the glomerulus, we have the filtration of water and dissolved substances, right? And then the filtrate travels into the proximal tubule. In the proximal tubule, all of these things are reabsorbed. And water, right? And then in the descending limb, oh heavens, hold on. In the descending limb, we have reabsorption of water. So that's like what we saw in this previous slide. So water is being reabsorbed into the bloodstream. And then in the ascending limb, we have reabsorption of some ions. And then in the distal tubule, we have some continuing reabsorption of ions, and then some more reabsorption of water and secretion occurring there. And then finally in the collecting duct. So remember, by the time we get to the collecting duct, basically you have your urine that will be excreted that travels through the medulla. A lot of water is reabsorbed into the collecting duct. And so water makes up the majority of your urine. This is kind of a cool table from your book. So essentially this is plasma means what started in your blood. Glomerular filtrate is what was filtered out in the glomerulus, and urine would be what you finally urinated out when it was all said and done after the whole extent of the nephron. So for example, sodium, let's say that your level of your blood sodium is 142. All of that was filtered out in the glomerulus into the tubular system, but you only urinate out 128 equivalents. That means that some of that was reabsorbed along the way. Potassium, plasma. So let's say our level's 5. All of that was filtered in the glomerulus, but we're actually going to urinate 60, which means that a lot of potassium was actually secreted, right, at the very end of the nephron from the bloodstream into the urine. One that I want to look at here is glucose. So let's say our glucose level was 100. All 100 units of that was filtered out in the glomerulus into the tubular system, and none of it ended up in the urine, which means all of it was reabsorbed into the body. Urea, our level is 26 in our bloodstream. Our kidneys filtered out all 26 units of that, but we actually excreted 1,820 units, which means that there's a lot of secretion going on as well. So this table is really good because essentially you can see what the levels were in the bloodstream. All of it was filtered out in the glomerulus, and then was it reabsorbed or was it secreted at the end? If it was reabsorbed, then this level is going to be lower than it was initially. If there was secretion, then this level is going to be a lot higher. So down here, you have a lot of your secreted substances, except for glucose. And then if that level was lower, then you have reabsorption. All right. So those concepts are a little complicated. I would advise you to read through your book. Essentially, though, I just care that you get kind of the skinny on that about basically just give me some examples of things that are reabsorbed or secreted. There's a lot of more detail in your book about like transportation and things like that. I don't care that you really know the details, but the overall concept is basically what I'd like you to have a good handle on. One more lecture 4.